My primary work is really has nothing to do with mites whatsoever, but like so many things in life, circumstances actually forced a kind of a capitulation into trying to figure out how to control these awful things. Most of my work, I'm an avian geneticist, and we're actually beginning to do some work in genetic resistance for H5N1 infections and other both low-path and high-path infections. And part of this work necessarily entails maintenance of breeding flocks of genetic lines that I have on campus at Madison. And even before I got my current position some 6 years ago, our poultry research laboratory, our facility on campus, has had a chronic mite problem that began actually 2 years prior to my arrival. We are still trying to eradicate these mites. And so part of what we've been attempting to do is to understand a little bit more about the biology of this little arthropod and seeing how, in fact, we can control it. So the northern fowl mite, just for some background information, is an ectoparasite that actually infests quite a bit of species of wild birds and also domestic poultry across the entire suite of species variation. Anything in close confinement can actually accelerate the rate and density at which these mites occur. They are hematophagous, which simply means that they are blood feeders. They're obligate blood feeders. They don't feed on anything else. The blood in itself is required for the mite to actually complete its life cycle. Now, because of their hematophagy, they can actually induce a significant amount of stress on the birds themselves. And this is not just in terms of acquiring blood itself, but it seems that it's possibly from immune system upregulates which actually interfere in a variety of other physiological functionings that the bird would normally endure without having these parasites on the birds. Most notably, what you'll often see in terms of heavy mite infestations is a sudden onset of anemia in the birds. And this also leads into lower egg production and also poor weight gain and a general susceptibility to other viral or bacterial maladies. These are all stressors. There's still a lot of controversy about what, in fact, damage the mite is causing, but at least people agree that these ideas in terms of loss of egg production, loss of weight gain, general anemic responses, in fact, are associated with mites, but it may be indirect through immune regulates. So not only are these mites kind of nasty toward the birds, but they can also affect humans too, not nearly as severely as one would see in terms of avian infestations, but in fact, it's usually the workers in the facility that would actually complain because while they won't infest a human, they do in fact bite humans. And in fact, you'll see a very, if you've gotten bit by one of these things, most people have a histamine response extremely rapidly, and it itches with the intensity of 1,000 white-hot suns. How many have ever been bitten by these mites? A few. They are extraordinarily itchy. And in fact, the folks at our poultry research lab, I mean, that's one of the biggest complaints. And so if you have mites in your operation, it would not only behoove you to kind of cure that issue for the benefit of your birds, but also your workers who might begin to have a worse attitude toward going into the houses. The northern fowl mite actually has an extraordinarily short life cycle of about an average of about a week under optimal conditions. Now I say on bird because all 5 stages of its life cycle do occur on the bird itself. Although adult mites are capable of surviving off the bird for up to about 3 weeks, after which they will in fact expire because of starvation. contrary to popular belief, the females, after blood feeding, they don't lay tens of thousands of eggs. And in fact, the females only lay on average about 3 or 4 eggs after each blood meal and may survive for months. So in terms of actual acquisition of a significant infestation, it actually is a fairly regular process, but it doesn't occur overnight. And so one of the things I'm going to advocate here is routine maintenance checks of your flocks, because if you catch it when in their early stages, it's actually possible to attenuate the problem very, very quickly. One of the problems in terms of control of this mite is the facet that it can live off the bird. And so what you'll often see are mites inhabiting the litter, particularly in pen flocks, or if they're outdoors, just in the general run areas. The problem there is that the mites actually now have a means by which to move. These things are actually very, very mobile. And so even if you have separate houses, like we do at the PRL, when we see an infection break out in one wing, our biosecurity prevents us from going from wing to wing. You can only go one wing at a time. We invariably get mites showing up in our adjacent wings. And we know, because of our biosecurity, that it's not through any there's not any reason for like people transferring equipment or food or anything like that. And in fact, we have evidence that we can see these mites leave the pens, they go on the cement floor and they walk. I'm not kidding. They walk, they will transfer themselves to a new house simply by walking there. And in fact, in cases, I've seen reports where severe infestations occur, that in fact there can be en masse migrations of millions of mites that are seeking new hosts. They just offload on the birds and they move, and they're attempting to find new hosts. I've been trying to get video of that. It has not been forthcoming, but it would be definitely worth a shot on television. Unlike their close counterparts, the tropical fowl mite, the northern fowl mites actually have a seasonal component to their infections. And despite what many people think, is that the mite infestations are typically more serious in the winter months and also in early spring. They seem to have an optimum temperature preference of around 65 to 68 degrees. And the seasonality is, in fact, somewhat fortunate in terms of actual mitigation through some of the control means, which we'll actually talk about in a few minutes. Typically, mites are first detected in the vent area of the bird. Again, there's some controversy about why you might find mites near the vent itself. Typically, I believe that in fact it's a highly vascularized area, both males and females. We also have large surface area of skin in which to locate themselves and to acquire their blood meals. And also it actually is a relatively humid environment, which the mites like, in contrast to the other portions of the bird. Other areas you'll see are on the saddle and also on the neck, and then particularly on the head. There is evidence that, in fact, they localize on the head and the vent because these are two areas that are more difficult for the bird to preen. And in terms of transmission of mite from bird to bird, having the mites present on the bird's vent area facilitates transmission from bird to bird, particularly if in your breeding flocks, not necessarily in your production flocks. Nevertheless, what you'll see in terms of kind of a mid-infection here is, if you look in the vent area, is that the mites tend to prefer young birds, on the average of 20 30 weeks, typically absent on old birds. And I suspect the reason is because the skin loses much of its elasticity, and then the mite actually has a much more problematic time acquiring blood on skin that has actually lost some of its elasticity. But what you'll see in terms of an earlier detection, this is actually, this is too far of a detection. This is already a problem. But you'll see localized concentrations of mites on the skin actually engaged in feeding. And when they're not feeding, they typically reside in the feathers. And they'll often sequester themselves between the individual barbs on the feather, which makes it more difficult for the host to actually remove the parasites from its body. Now, as these populations increase, the severity of the infection certainly becomes more visible. So in early stages, we see here, we see a reddened area, which would normally be pink or a lightish color. This isn't due to inflammatory response of the bird, but you can see here that already we're starting to get some skin sloughing and probably some carcass that have been shed from the mites. And in advanced infections here, what you're really seeing here primarily, in addition to things like dried blood, the cast skins of the mite, is actually, these are mite feces here. And so this is a very advanced stage. And you can almost imagine from the amount of fecal material that's being produced by an animal, that's about 0.5 millimeter in diameter. This is a huge infection here. And even though that female will lay only on average 2 to 5 eggs after each blood meal, a very young bird can quickly achieve kind of a mid-level infection of somewhere around 20,000 of these mites per individual bird, distributed primarily in the vent region, also the neck and head area. And that can occur in as little as 2 months. So if you ever see a mite on your bird, you have to take immediate action. Because in heavy infestations like you see in this stage, in fact, the calculations have shown that an adult bird loses about 6% of its total blood volume to mites feeding per day. It's something that you cannot ignore. In terms of control, primarily chemical control, and in fact, it actually works pretty good. You have three, I should say you should have about 3 primary chemical controls. First, you have the pyrethroids like permethrin. I should say these are all neurotoxins. Next, we have the organophosphates here. Tetrachlorovinophosph, dichlorovoss, or simply RAVAP. This is the one we actually use at the PRL, and I think most commercial facilities will actually use RAVAP. We also have malathion and carbaryl. Together, these are all cholinesterase inhibitors, and as such actually have a high degree of toxicity associated with them, which makes their use somewhat questionable despite their effectiveness, at least in terms of the organophosphates and the carbamate, carbaryl. Lastly, a very effective, and in fact it's actually extraordinarily effective, is depopulation of the wings. Although for most facilities, this is just not a viable option. And I believe that this was one of the reasons that we're having problems at the PRL, is because we used to be able to depopulate the entire facility, and if you can do it for more than about 21 days, which is about the maximum time that the mite can actually live off bird, you can actually return the birds to a clean house. But that's not a viable alternative for most, and it's certainly not even viable for us since Arlington had been closed down a few years ago. Well, while these chemicals do seem to be, in fact, quite effective, at least in terms of the pyrethroids, the mites are known to actually be able to generate resistance to application very, very quickly. Carbaryl is actually being phased out currently because of environmental concerns. And the remaining organophosphates that we have actually have acute and chronic toxicity concerns, not only for the birds themselves, but also for the applicators of this. And there's a peculiar regulatory issue that pops up, is that since these types of organophosphates are in fact termed pesticides, they're actually regulated by the federal EPA and not USDA. So in terms of USDA, any veterinary staff cannot legally go off-label in order to perform the administration of these organophosphates. And so you're bound by the label directions, which, as I'll point out to you in just a few slides, is not necessarily the best way to go, but nevertheless it is what you must abide by. Because of this, we've also found that, now this probably doesn't affect most of you in here, but in terms of the toxicity, not only are we talking about the humans and the birds themselves, but we also found that these organophosphates were interfering with some experiments that we were performing. One of the things we do is looking at, we use birds as a model for fetal alcohol syndrome in humans, and we found that application of the organophosphates just destroyed all of the results that we did. And we found this purely by chance, when things would work for a while and then they wouldn't work, and then they worked and then they didn't work. And we found, in fact, just by chance, that in fact it had to do with the periodic application of these organophosphates. So in terms of a research goal, that let's see if we can find an alternative to these organophosphates. Well, one of these is a biocontrol agent. It's actually an entomopathogenic fungi named Beauveria bassiana. It's actually a common soil fungus that's ubiquitous around the entire planet. And it actually naturally infects many arthropods, such as ticks, flies, even beetles. And the effective portion of the fungus itself, it's actually an ascomycetes fungus, are the spores, or more precisely conidia, which will actually, when they land on the insect, I got 3 examples here of some North American species. Some of you might recognize this one in the middle as a Colorado potato beetle, nasty little thing. But in any sense, once the spores land on the insect, the spore will germinate, send out hyphal strands that actually produce very potent chitinases, which is essentially they bore through the exoskeleton of the organism and began to proliferate internally. And in addition to growing quite rapidly throughout the internal organs of the insects, they actually have the notoriety, the fungus has the notoriety of actually producing antimicrobials that actually inhibit gut microbes from competing with it. So essentially, in a very, very short amount of time, the Beauveria can attack the insect, proliferate throughout its interior components, and then resend out the fruiting structures, the conidia, again externally, to continue the life cycle. Now, some entomologists actually recognized this and thought, well, we could use this as a biocontrol in terms of our own poultry flocks. And so the way that works is that presumably a lot of undergrads or graduate students go out, take soil samples, isolate the fungus, and then treat the target organism to see if in fact it is effective in terms of killing it. And so recently there's been quite a surge in this, because there is no toxicity associated with the Beauveria, with this fungi, at least any that are known to humans. And it's actually very effective and can kill these insects, the target hosts, very, very, very quickly. Well, we wanted to see, is there anything worth looking at here? like so many products, much of it can actually be a lot of talk, and then when it actually is applied,